But what we saw was that the dogs that were fed once a day had a lower risk for all 9 of the age-related conditions that we looked at, and tended to have better cognitive function.
The goal of the Dog Aging Project is to understand how genes, lifestyle, and environment influence aging, and we want to use that information to help pets and people increase health span. So can you tell us a bit more about the Dog Aging Project and what we've learned so far?
Sure. So the Dog Aging Project is a large, what we call longitudinal study of aging in companion dogs or pet dogs. And there's really 2 major goals to the Dog Aging Project. The first is to understand what are the most important genetic and environmental factors that influence healthy longevity in companion dogs. And the second goal is to do something about the biology of aging and to determine specifically whether or not the drug rapamycin can increase lifespan and health span in companion dogs. So the largest component of the Dog Aging Project falls in that first bucket, which is really about understanding the biology of aging. What are the genetic and environmental factors that influence aging in dogs? And we have about 41,000 dogs now in the Dog Aging Project longitudinal study. And in that study, we're actually not asking owners to do anything different than what they would normally do. So we collect survey information from the owners, veterinary medical records, and we update that information annually, or we ask the owners to update that information annually. And then a subset of those dogs, we have biological samples collected from. So metabolome, microbiome, epigenome, genetic sequencing. Again, and the goal there is really to try to integrate as much of that information as we can to pinpoint the correlations that are most strongly associated with lifespan and health span in dogs. I do think it is important to state, and I'll probably say this more than once today, that from a longitudinal study, an observational study like this, we are limited to correlations, right? So we can't actually determine causation when we identify these factors that are associated with health outcomes during aging. So it's really a very large hypothesis generating study. The clinical trial, which we can dive into a little bit more deeply if you'd like, is a true randomized, double-blind, placebo-controlled clinical trial where dogs either get placebo or rapamycin, and we're really trying to understand and determine, does rapamycin increase lifespan and health span in dogs?
So should we start with the correlations first? Has anything popped in the data that you've collected so far as a correlation to aging?
Sure. So the answer is yes, and I'll give you a couple of interesting examples. I also want to make the point that this first data set, so a couple things to say. The Dog Aging Project is an open science project, so we have actually released our first year's data set to the scientific community. We published a paper in Nature earlier this year, sort of describing that data set and the design of the study. So if anybody out there is a data scientist and they love dogs and they're interested in aging, this I think is a great resource for people to dive into, and it's really easy to get access. Just go to the website dogagingproject.org. There's a little button there that says request access to the data. Very simple data release form. So just want to make that appeal for people to come and use this data. So the reason why I'm talking specifically about this first year's data set is because that is what we call a cross-sectional data set. So we are a longitudinal study. That means following the dogs over time and collecting repeated data on those dogs every year. But this first year was, because it was the first time point, it was just one snapshot in time. So it's what's called cross-sectional. So that's a limitation to this first data set that we've analyzed. But even still, we've identified some, I think, interesting observations. One that I was particularly interested in was the association between diet and longevity and health outcomes during aging. Pretty much anybody who's been around this field at all is aware that caloric restriction is in many ways the gold standard for an intervention that seems to increase lifespan and slow aging or improve metrics of health span in laboratory animals. And And that's crystal clear, rock solid. We actually might want to talk a little bit about that because it gets overinterpreted, I think, in terms of the magnitude and consistency of the dietary restriction or caloric restriction effect. But lots and lots of studies showing that caloric restriction can increase lifespan in rodents, at least, and in invertebrate models. But more recently, there have been this sort of proliferation of what I call variants of caloric restriction or dietary restriction, like time-restricted feeding, intermittent fasting, ketogenic diet, protein restriction, all that stuff. I sort of lump under, a variance of dietary restriction. And I thought dogs might be sort of an interesting opportunity to look at something like intermittent fasting or time-restricted feeding in the real world, because owners often feed their dogs a different number of times each day. So some people, in fact, most people feed their dogs 2 times a day, but there are plenty of people who feed their dogs once a day, people who feed their dogs 3 times a day. There's actually this percentage of the population in the United States, or at least in our cohort, I think it was around almost 10% of people feed their dogs ad libitum, meaning the dog has free access to food. There's a self-selection there. There are some dogs, if you give them free access to food, they'll blow up and turn into obese dogs. But in any case, it's sort of this natural population where owners have, for a variety of reasons, selected a different feeding frequency. And so we just asked the very simple question, was there any correlation between the number of times each day that a dog is fed and prior diagnoses for a variety of age-related disorders, when we also looked at cognitive function. And I really didn't expect to see anything, because I have been unimpressed, I would say, with the time-restricted feeding data in the literature. But what we saw was that the dogs that were fed once a day had a lower risk for all 9 of the age-related conditions that we looked at, and tended to have better cognitive function, or at least a reduced risk of dementia. And in, I think, 6 of the 9 cases, it was statistically significant. So the directionality was the same in every case, and it reached significance in several of the cases. So, that's consistent with the idea that time-restricted feeding, or at least once-a-day feeding in dogs, may have beneficial effects for a variety of age-related health outcomes. But again, I think it's super important to emphasize that this is a correlation, and there are some pretty obvious potential explanations for that correlation. The one that I think is probably most likely to be the case is that dogs fed once a day are less likely to be obese, and so it may actually be the obesity or lack of obesity in those dogs that is causing them to have a lower likelihood of a prior disease diagnoses in these categories. Nonetheless, I think it clearly suggests some specific hypotheses that can be tested in clinical trials going forward.
Did that have any correlation to do with whether the dogs were fed in the evening or the morning? Because there's a big hullabaloo at the moment about some of the human research maybe suggesting that actually eating within the smaller window in the day, but so long as it's in the morning, may actually have some beneficial effects.
Yeah, so we don't have that data. So we are limited at this point by the way that the survey instrument asks the question. So the original variation of the version of the survey instrument did not specify time of day that the dogs were fed, just how many times each day. It's an interesting question, and I think, again, something that we can look at going forward. Unfortunately, we don't even really have information on whether the dogs were obese or not. We have body weight, but there isn't a correlate of BMI in dogs that works very well. I mean, BMI doesn't work perfectly in people. It's even worse in dogs because of the wide variety of body shapes and sizes in dogs. So we don't really even know whether the dogs were underweight, normal weight, or overweight, or at least obese. So I think there's lots of questions. This idea that the window, that it's both the window of time and the time of day that you eat, I think is super interesting. And there's some really important biology to uncover there. I will also say, though, that, from my own digging into the literature, I think there's real effect there, but it's very, very small. So when you look even in mice, it turns out that there absolutely is this interaction between circadian rhythm and diet and caloric restriction and time-restricted feeding. But the vast majority of the effect on lifespan and health span comes from the caloric restriction part, not the circadian part. At least that's what the literature right now says. Obviously, that may change in the coming years, but my impression is that really what's most important is what you eat and how much you eat, and when you eat might add a little incremental effect on top of that. But if you don't get the what you eat and how much you eat part in line, I don't think it's going to have any effect.
Yeah, that's pretty much what I tell my patients as well, that if they're going to focus on anything, just make sure that they focus on actually what they're eating and how much they're eating. Some patients, though, they do find it useful to restrict their calories through intermittent fasting, and in which case I think that's great. But on the point of diet, have you noticed any correlations with the types of foods that the dogs were eating in terms of health span and lifespan?
So we're just looking at that right now. And again, I would say, so the answer at this point is we've done the initial analysis. We haven't published it yet, so I can't really dive into the details. What I can tell you, though, again, is we're somewhat limited by the survey instrument itself and what it asks. So the kinds of things we can look at are, comparisons of raw food, cooked, homemade food, dry kibble, things at that sort of resolution. We don't have yet food composition, down to a more granular detail, or even brand of dog food for the commercial food. So we have commercial versus homemade. We have raw versus cooked. There are some differences, I will tell you that. Nothing as large as the effect that we saw correlated with frequency of feeding, though. So my feeling right now from what I've seen of the data is that frequency of feeding has a much greater, a much stronger association, I would say, with age-related health outcomes than composition of the diet at the level of resolution we have for composition of the diet, which isn't particularly fine-grained at this point.
So we've talked a bit about diet. Has anything else come in terms of correlations from the data that you've already accumulated?
Yeah, the other one that I think is particularly interesting was the relationship between activity and cognitive function. So that's not shocking. I mean, it's not shocking that there would be a relationship. We have pretty good evidence in people, right, that exercise is associated with a bunch of improved health outcomes during aging, including reduced risk of dementia. We very much see the same correlation in our data set. So this was a study looking specifically at factors that were associated with risk of canine cognitive dysfunction, which is the sort of clinical bucket that all canine dementias get put in. And the strongest predictor or the strongest association with dementia was activity. And so the dogs that were most active, and again, very low resolution measures of activity. So this is owner reported, highly active, moderately active, low active, sedentary. But when you look between sedentary and highest reported activity, I think it was about a 7-fold difference in risk of a diagnosis of dementia. That was second only to age. I mean, not surprisingly, age is the strongest risk factor for dementia and pretty much every other age-related disease. But second to age was activity and exercise. So again, nothing super shocking there, but it's nice. It's kind of a sanity check. And it's nice to see in this, socially relevant large mammal that we're seeing the same sorts of relationships between activity, exercise, and dementia. And I anticipate that we'll see similar beneficial associations between activity and other age-related outcomes in dogs as well. There's some other interesting stuff going on with cognitive function in dogs. So the tool that we are using right now to assess dementia in dogs is a survey-based instrument. So it's a very short, takes owners like 15 minutes to complete it. It's been clinically validated as a diagnostic tool for dementia in dogs. But it's been used as a binary tool. And what I mean by that is that the clinical validation is above a certain score. So when you add up all the answers, if the score is above 50, that is diagnostic for dementia. Below 50 is no dementia. So it hasn't been proven to be useful as a continuous tool. In other words, would a score of 45 be indicative of mild cognitive impairment, or a score of 40? So we're very interested in answering that. It looks like it can be. So across our data set, I think we've got certainly more than 30,000 cognitive assessments now on the dogs in the longitudinal study. Very much looks like it can be used as a continuous tool. You see the same age-related decline in cognitive function that you would expect to see. Looks very much like in people. There's a couple things that I think are really interesting in that data set, though. One is, at least at a high resolution level, there doesn't seem to be a sex effect. So in other words, female dogs don't seem to be at an accelerated risk of developing dementia with age, or at least cognitive decline with age, compared to male dogs. That's interesting, because there's this, certainly in people, more women have Alzheimer's disease than men. And there's this idea that women are at higher risk. So that's interesting. The thing I thought was super interesting, though, is there isn't a body size effect. In other words, when you look at giant dogs, large dogs, medium dogs, small dogs, there doesn't seem to be an accelerated risk of dementia as a function of age in the larger dogs. Now, that's interesting, because in every other parameter that I'm aware of, and the ones that we've looked at, big dogs age faster than small dogs. So they're at higher risk of cancer and kidney disease and heart disease with age. That doesn't seem to be the case for cognitive decline and dementia. So, I think that's telling us something probably important about the biology of the aging of the brain, maybe relative to the biology of aging in the rest of the body. And so I'm super interested to see where that research goes as well.
Just to tie off the dog aging project, so you mentioned rapamycin, which, we've designed a clinical study in humans to look at, but what are we hoping to achieve with the dog aging project by looking into rapamycin?
Right. So, the goal of that clinical trial, as I alluded to earlier, is really to try to answer the question, does rapamycin increase lifespan and health span in companion dogs? And I think, because dogs age about 7 to 10 times faster than people do, it's possible to actually do a healthy lifespan study in a reasonable amount of time in dogs. So our primary endpoint for this clinical trial is lifespan. And the dogs have to be of normal health status when they come into the study. So they have to be at least 7 years old, so middle age. They also have to be at least 40 pounds, because, as I just said, big dogs age faster than small dogs. So in that demographic group, middle-aged, larger dogs, with 580 dogs, that's the size of the study, half placebo, half rapamycin, we're statistically powered to detect a 9% change in lifespan over 3 years. So that's something you just simply couldn't do in humans, right? You can't do a healthy longevity study in humans. Well, let me take that back. It would be extremely expensive, and feasibility would be challenging to do that study in humans. In principle, if you enrolled tens of thousands of people into a study, you could do it. But financially, it just isn't reasonable to expect anybody's ever going to do a healthy longevity study in people. But we can do that in dogs. So the goal is really to answer that question. Does rapamycin increase lifespan in companion dogs? But of course, health span is, as important, some would argue more important than lifespan. So as secondary endpoints, we're looking at many different health span metrics to really try to also address whether rapamycin is broadly improving health during aging in companion dogs. And we're looking at many of the same health span metrics where rapamycin has been shown to delay or even reverse functional declines in mice. So that's the incentive for doing that trial. We're well on the way to enrollment. We're still enrolling dogs into that trial. It's been, as you might imagine, it's been challenging. Lots of challenges go along with running a clinical trial. This one's no different, but we're well on our way. And my hope is that we will be fully enrolled. I mean, I would love to say by middle of next year. I think realistically, I keep changing the date, unfortunately, but I feel like we're on track to complete our randomization by the end of next year at the latest. Still crossing my fingers we can do it by middle of next year. And then once all the dogs are randomized, it's a 1-year treatment period, 2-year follow-up. So, we'll know the answer in probably 4 years. I'd love it to be faster than that, but realistically, I think that's probably where we're at. Still, that's a pretty good time frame considering, the outcome of lifespan that we're looking at in this study.
I think it'll be quite interesting just for the viewers to go through the mechanism as to why we think rapamycin may actually improve health span and lifespan. So do you mind just speaking on that?
Sure. So there's two things I would say. One is the mechanism. I think we can dive into. I'll just leave it at rapamycin is a specific biochemical inhibitor of a protein called mTOR, which stands for mechanistic target of rapamycin. Why we think it's of interest in the biology of aging is really based on the massive amount of data in the literature showing that if you inhibit mTOR, either genetically or with rapamycin pharmacologically, you increase lifespan and appear to broadly slow the molecular and functional declines that go along with aging in every model organism where it's been tested. And that includes yeast, which is a single-celled model organism, nematode worms, fruit flies, and mice and rats. And I think in mice and rats, for me, the data are particularly compelling because rapamycin has been shown to increase lifespan by many different labs multiple times across a very wide dose range. So it's sort of unique in the geroscience / longevity field for that reason. It's really the most consistent intervention at extending lifespan that we have. In fact, it's even more consistent than caloric restriction. Magnitude of effect isn't quite as big as caloric restriction, but consistency seems to be better. And importantly, not only does it extend lifespan, but I think in every tissue where people have really looked carefully, there's evidence for either delayed age-related changes at both the molecular and the functional level. And what's most impressive to me now are the multiple tissues and organs where rapamycin treatment, starting in middle age, actually seems to reverse functional declines partially. I'm not saying it makes them go all the way back to a youthful state, but functionally you can see improvements in an aged animal in heart and immune system and oral cavity and in female mice in the ovary. So, it seems to be a very robust, reproducible intervention for modulating the biology of aging in a way that leads to increased lifespan and, at least to the extent that we can measure it, improvements in multiple metrics of health span, even when the intervention is started in late age. The last thing I'll say, so I mentioned started in late age. That's been shown, again, by multiple groups. You can start the treatment at about, the biological equivalent of a 60-year-old at mouse, even later sometimes. Still see most of the benefits. And there's emerging studies now that even transient treatments or cyclical transient treatments with rapamycin can have many of the same benefits. So, that's particularly, I think, compelling from a translational perspective, because those are the kinds of interventions we would want to think about moving into the clinic, whether it's a veterinary clinic or a human clinic. Those are the kinds of translational geroscience interventions that I think have the most potential to be developed, because it doesn't require starting at a young age, may not even require taking it continuously starting in middle age. Maybe you can get many of the benefits from, shorter term treatments where the likelihood of side effects are going to be reduced compared to something that you maybe would have to take your entire life. So that, again, I think is another benefit to rapamycin, at least given what we know about from the preclinical studies at this point.
So if we explore the study design that we've come up with for our rapamycin study in humans, where we're taking older adults and we're giving rapamycin once a week and we're exercising them the other times in the week. So what we're trying to do is have periods of time where we're actually switching mTOR on via exercise, and then separate periods of time where we're switching mTOR off. So I just wondered if you could speak to that balance, because taking, for example, people trying to restrict protein intake, because protein also switches mTOR on. Yet we've got these, very strong preclinical studies showing that if you seem to switch mTOR off with rapamycin, that extends lifespan. So what do you make of the mTOR balance? And at the moment, knowing the current research that we've got, how should people be trying to strike that perfect balance?
Yeah, I mean, I think this is a really good question. I also feel like there's very little good evidence out there to argue for a specific sort of, regimen of rapamycin in the context of exercise. That's why I think your clinical trial that you've proposed is extremely important. So maybe it makes sense to take a step back and maybe start by talking a little bit about mTOR and its role in muscle function, muscle mass. So if you look at the muscle biology literature, for many, many years, we've known that activation of mTOR is necessary for muscle hypertrophy, muscle growth, right? So the prediction in the muscle biology community was that rapamycin should lead to muscle loss, right? And so I think that if you had asked a typical muscle biologist 10 years ago, what would happen to people or mice taking rapamycin, they would have said it will accelerate sarcopenia, which is muscle loss with age, and lead to a decrease in muscle mass. The outcome, that experiment's now been done multiple times in both mice and rats, and the outcome is exactly the opposite of that prediction. So rapamycin treatment in the context of normal aging actually seems to preserve muscle mass. So it prevents or at least attenuates sarcopenia. That's crystal clear. Unfortunately, I don't think the muscle biology community has caught on. They aren't really paying attention to that literature because you'll still see this nonsense about how rapamycin is going to cause muscle loss. And I don't think there's any evidence to support that either in the preclinical literature or really in the clinical literature. So we have more than 20 years of evidence now for people taking rapamycin clinically, mostly in the context of organ transplant patients. I'm not aware of any evidence that organ transplant patients taking rapamycin show muscle loss compared to organ transplant patients not taking rapamycin or mTOR inhibitors. So I don't think there's any real evidence that at the clinically useful doses, rapamycin leads to lower muscle mass. And there is clear evidence, again, at least preclinically, that rapamycin attenuates muscle loss with age. Okay, so that's not exactly what you were asking, but I think it's really important to put this in context. So a different question would be, would rapamycin prevent muscle gain if you were to embark on a resistance training regimen with optimal diet? That I don't know the answer to. It wouldn't surprise me if taking rapamycin, say once a week or at organ transplant doses, it wouldn't surprise me if it did somewhat attenuate the ability to put on muscle mass if you were to really try to grow muscle. But it wouldn't surprise me if it didn't. It also wouldn't surprise me if it actually helped. So I think we just need to know, right? We don't have any data to support or refute that hypothesis. What I can tell you for sure is I know many people who have been successful at increasing muscle mass, increasing lean mass, reducing fat mass while taking rapamycin. These are people who are middle-aged, between 50 and 70 years old. So what I can say with certainty is the doses of rapamycin that most people are thinking about for, sort of potential health span purposes are not incompatible with gaining lean mass and reducing fat mass. Does it slightly attenuate the ability to gain lean mass? I don't know. We need to do the studies. But I would be very surprised in your clinical trial. Again, this is my prediction. We need to do the studies. I'd be very surprised if rapamycin had a significant negative impact on ability to gain muscle mass. I would be shocked if rapamycin led to a loss of muscle mass relative to baseline. I don't think there's any reason to think that's the case. So, and we could talk a little bit more about the potential mechanisms and all of that and timing. I think it is an interesting question. So if you were to, if you could figure out when is the optimal time frame after resistance training for mTOR activation to build muscle, you could potentially rig the system so you're taking rapamycin at a time or frequency that doesn't inhibit mTOR, at least theoretically, in that context. And I know you've thought about that in your trial design. Again, my My bias is that biology is way more complicated than we understand, and I don't even know that would make a difference. I also think it's really important to appreciate that when somebody takes a rapamycin pill, the effect of rapamycin on mTOR is different in different tissues, and the dose of rapamycin necessary to inhibit mTOR in different tissues varies, right? So I'm not even sure that the doses of rapamycin that people are taking now would have would significantly impair mTOR activation following resistance training. I think that we would need to know. We would need to actually look at that. There have been a couple of very short-term studies looking at that I'm aware of in people, but nothing particularly convincing. So the real answer is I don't know. I mean, I'd love to know the answer. I hope that you can get your clinical trial funded, because it would be nice to actually have data instead of, people just sort of speculating based on their bias and usually a lack of informed opinion of the literature about what the effects would be. It'd be nice to actually have real data and say, okay, here's what we saw in this clinical trial.
So on that point, shameless plug, if you are in a position to donate, there is a donation link in the video's description. But coming back to mTOR and protein restriction in particular, so you wrote a fantastic review article, I believe it was last year, titled Anti-Aging Diets, Separating Fact from Fiction. So trying to tie this into mTOR. So there's a couple of mice studies suggesting that if you restrict protein, that seems to result in longer lifespan in some mice strains. And the presumed mechanism is that there's maybe slightly less activation of mTOR. And that's led to some people suggesting that we should be restricting protein intake. Now, in my clinical experience, that's the last thing that you want to do. I've been really encouraging my patients to eat more protein, and I've been encouraging viewers of this channel also to eat more protein, so long as it's lean protein sources. So I'm very curious to hear your opinion about protein intake, what the preclinical research actually shows, and what you personally do with protein.
Right. So again, I think we have to be honest that there are no concrete answers here, right? So what I can tell you from the mouse and rat studies is there are multiple studies showing that if you restrict protein in the diet to very low levels, that can increase lifespan, at least in some genetic backgrounds, not all genetic backgrounds. It's interesting there because the mice actually, it depends on the study, whether they're also calorically restricted. There are actually studies showing that with certain protein restriction regimens or with branched chain amino acid restriction, so just restricting some of the amino acids that are known to activate mTOR, you actually get mice that eat more than the controls, but yet they don't put on weight. So they're leaner in all cases, or at least lower body weight. Let me take that back. They're lower body weight in all cases, but they're actually eating more. So it's complicated biology again. The other thing I would say is it's not really clear that the longevity effects from protein restriction or branched chain amino acid restriction are only going through mTOR. So again, I think it's really important to appreciate that all of these dietary interventions are affecting thousands of chemical reactions in cells and tissues. So there are lots of things happening at the cellular biochemical level, at the systemic hormonal level when you substantially restrict any macronutrient, or if you do true caloric restriction. And the effects of those changes in combination with mTOR inhibition could be different than the effects of something like rapamycin, which is really very specifically inhibiting mTOR and in most cases not having those broad-based changes, to thousands or tens of thousands of chemical reactions. So I think it's just important to appreciate that it's probably not only mTOR, and we don't really know how much of the physiological effects of protein restriction in rodents is due to inhibition of mTOR versus other effects of protein restriction. But it can increase lifespan. So there is this, at least some people have argued that we should restrict protein in people because if it extends lifespan in mice, maybe it would extend lifespan in people. My personal view of that is that just doesn't make sense. Given what we know about the importance of maintaining muscle mass with age and the problems that go along with losing muscle mass and frailty with age in people. So I think this may be a case where the preclinical studies can be, the mouse studies, when I say preclinical, I mean in this case mouse and rat studies, can be very useful at identifying the specific mechanisms by which protein restriction is engaging the longevity response. I am very skeptical that that intervention itself in people is going to be beneficial. And I actually think this may be a case where we need to be really careful what we say, because I'm very concerned that protein restriction will actually be harmful for many people, particularly people who are middle-aged and above. And so that's, again, it's a guess. I don't have a lot of data to support that. There are some epidemiological studies supporting the idea that low protein diets after about the age of 60, I actually think it's probably closer to 50, but the epidemiological studies that I'm aware of certainly say that after the age of 60, a low protein diet is associated with higher all-cause mortality. And what I mean by that is people who eat a low protein diet tend to die earlier once you get past age 60. So there is evidence in people that a low protein consumption is associated with higher risk of death. But I just also think that everything we know about the importance of preserving lean mass and function as we get older suggests that we don't want to do anything to impair that. And I would worry that a low protein diet is going to impair that. So, my personal belief, and my diet matches this, is that working hard to at least maintain, and I would say build muscle mass when you're in your 40s, 50s, 60s is exactly what you want to do. And that requires at least a moderate intake of protein. The last thing I'll say on this as well is the guidelines for protein. There's a lot of lack of understanding out there about what the guidelines for protein actually are. So it's my understanding that the RDA, the recommended daily allowance of protein, I actually went back and looked at the paper, so I apologize if I'm misstating this, but my understanding is the way that was developed was the minimum amount of protein that a group of sedentary young men needed to eat to avoid negative nitrogen balance, which would mean loss of muscle, right? So the recommended amount is really a minimum amount, and so what we call high protein, low protein, that's relative to what your baseline is. If your baseline is the RDA, I would say that's low protein. And that's what we should be thinking of as a low protein diet. You probably want to be substantially above that. That's my guess. I'd really like to see better, more modern studies. I also think, we need to know what's the difference between sedentary, moderately active, very active people in protein needs, the minimum protein necessary to avoid muscle loss. We also need to know what the impact of age is on that and what the impact of sex is on that. So I think there's a lot of work to be done to actually get us closer to a recommended protein level where you wouldn't be expected to lose muscle mass. The last thing, I just said that what I just said would be the last thing. This is actually the last thing I'll say on this. I also think it's important to appreciate that a high protein diet, which is, I guess my diet relative to what people think of as, low, moderate, high would be considered high protein. I don't think that the risks are substantial from a high protein diet if you are not overweight and obese and if you are active. I think the consequences of a high protein diet in terms of health risks are much greater in people who are sedentary and overweight. So again, I think it's really important to get the foundation of your health put together before you really worry a lot about the specific macronutrient composition of the diet. Although obviously macronutrient composition is going to impact your ability to get to a healthy weight if you need to get to a healthy weight. But I do think it's important to appreciate that the risks of a high protein diet may be very different in overweight and obese sedentary people than they are in healthy weight active people. And most of the epidemiological data comes from the general population, which, as we all know, at least now, is mostly overweight, obese, and sedentary. So those are important things to consider when you look at the epidemiology as well.
Yeah, it's just the the patients that I see in the clinic, generally they're either middle-aged or older, and one of the biggest markers of frailty that I see is loss of muscle, where patients don't have the strength to, look after themselves, to shower themselves, to get up and down off the toilet. And it's those sorts of things that I do get extremely concerned about this protein restriction idea. So I'm fully in agreement with you. And also speaking with these older people, often they're just eating toast and tea for dinner, and they're not actually having, their protein intake. I spend a lot of time in the clinic now actually talking to them about diet, about how you do want to be having protein, and trying to a little bit dispel, not the myth per se, but the fear around some protein intake. We've talked a lot about rapamycin. But from the interventions testing program, which is a program that looks to identify agents to extend lifespan and health span in mice, we've got a recent study coming out looking at a combination of rapamycin and acarbose, which extended lifespan by about 29%. So aside from rapamycin, what agents are you quite excited about the potential to extend health span and lifespan?
Sure. So I think there's a few things I would say on this. One is I think that acarbose plus rapamycin study was very interesting. It suggested that there may be sort of an additive effect between rapamycin and acarbose when you combine them. Certainly not definitive for that interpretation, though. And a couple things make me say that. One is that the individual controls were not in the same study, so that limits interpretation. But you can look at the magnitude of effect and say it does seem pretty likely that the combination of acarbose plus rapamycin, that the dose of rapamycin that they used, is bigger than that dose of rapamycin by itself, and certainly bigger than that dose of acarbose by itself. But, and this is, I think, an important but, the dose of rapamycin that they used in that study was not even the optimal dose that the ITP has tested. So first of all, I think it's important to appreciate, we actually don't know the full dose response for rapamycin for longevity in mice. But the ITP has actually shown that higher dose of rapamycin in mice can extend lifespan by more than the dose that they used here. And in fact, that dose, the high dose of rapamycin, is comparable to rapamycin plus acarbose in this study. So the interaction there, I think, still remains to be determined. But I do think this idea of pairwise combinations is important to look at in longevity studies, because we have very little in the way of data for even combining two different interventions and seeing what the outcome is. So that was a nice component of this. So your question, though, what molecules am I excited about? This is going to disappoint people, but I'm not really excited about a whole lot, to be honest with you. Rapamycin works, no question about it, in mice and rats. I believe. In fact, I am personally convinced that rapamycin can have beneficial effects in people. I don't know if it's going to increase lifespan, but I know many people who appear to have had beneficial outcomes from taking rapamycin during middle age. So I think there's something there. The other drugs that we know about that in mice that extend lifespan are all smaller than rapamycin. They're all either they're sex specific and smaller than rapamycin, meaning they only work in males or females, or they're just smaller than rapamycin, but they're all smaller than rapamycin. So what does that mean? I mean, I think that those, and I would say we haven't done full dose responses on most of the interventions that the interventions testing program has found can increase lifespan in mice. So I think that's kind of the next step. I would like to know, can any of these interventions work as well as rapamycin? I see a lot of people who there seems to be this perception in the, maybe this is just a Twitter longevity community, and I don't even want to talk about Twitter, but there seems to be this perception that, as soon as the ITP shows that something works, we all have to start taking it. That's ridiculous. Most of the stuff that the ITP has shown can increase lifespan in mice is like 4%, right? And at one dose. So we don't know much about the biochemical mechanisms in most of these cases, the dose response, and in many of these things only worked in male mice. So I am very much of the feeling that we need to learn more about how these things are working before there's this rush for people to start taking them. So in that context, I guess I'm not particularly enthusiastic about a lot of the interventions for human consumption now. I'm very enthusiastic about what these interventions might be able to tell us about the biology of aging and the mechanisms that we can target to impact the biology of aging. I think some of them, make sense, right? So we've got these, so acarbose is a diabetes drug. Canagliflozin is another one that the ITP has shown can increase lifespan, reduces blood glucose levels. So we know these things have clinical utility already. We know something about the side effect profiles in people who have glucoregulatory challenges. In particular, those drugs might be useful. Metformin I would also put in that class, although metformin is a very different and interesting case example where it doesn't actually seem to really extend lifespan, at least not consistently in mice, but it is an anti-diabetes drug. So those things I think are potentially interesting, but I wouldn't rush out and start taking them, especially if you don't have any glucoregulatory impairment. I don't think it makes a lot of sense to start stacking these things until we know more about how they're working, what the dose response might look like in people. That's my personal opinion. Like, I know there's lots of people out there who are in the biohacker crowd and they want to do these sorts of self-experiments. That everybody's got their own views on the risk reward. But my personal opinion is I'd really like to see something work better than rapamycin. That's what I'd really like to see. And I don't feel like as a field, we are working that hard to find those things right now.
I suppose the counter that I'd give to that is the SGLT2 inhibitors. So you've mentioned the canagliflozin. And this is just based on my clinical experience here in New Zealand, we use empagliflozin, and that's fantastic in heart failure, and is actually now, so you don't even have to have diabetes, and you get these heart failure benefits. It seems to be the same thing with chronic kidney disease, particularly if your kidneys are leaking protein. Again, if you don't have diabetes, this medication seems to be brilliant for that. And I just wonder whether this class of medication will actually be one of the first to be used in humans to slow down kidney decline. Our kidneys start to decline from about the age of 30 to 35. And I do wonder whether this class of medication will be the first one to actually slow down that process. So I certainly don't advocate that people should use it yet outside of those clinical contexts that I've mentioned. But I do find that the potential of this class of medication to be very exciting. What do you think about that?
Yeah, so I think, you've got clinical experience that I don't have, right? So I think that's a compelling sort of argument in potentially, certainly in favor of testing these things more deeply. So what I would say about canagliflozin is, if you look at the data from the interventions testing program, I think they've tested 3 doses now. And the biggest effect was around, I think, 14%, and that was only in male mice, right? So again, from the perspective of the biology of aging, we don't really understand why it only works in male mice, and it's about half the magnitude of effect that can be achieved with rapamycin, right? So that's what I would say about the comparison of those, and what I was talking about before about I'd really like to see something that does better than rapamycin. I think the human, and metformin you might put in this bucket as well. Metformin is, largely, in my view, unimpressive in the mouse studies, and yet there are lots of people who find the clinical data in people compelling, suggesting that metformin may provide protection against a variety of age-related diseases, at least in people who have diabetes. Metformin also, I think, is potentially an interesting cautionary tale because, there was this study that came out a few years ago now that reported that that diabetics taking metformin actually had reduced all-cause mortality, certainly compared to diabetics not taking metformin, but they suggested that even compared to non-diabetics not taking metformin, that diabetics taking metformin had lower all-cause mortality. There was just another study that came out that found the opposite, right? So I think we have to be very careful about extending the use of these medications in populations where they have a disorder or a disease and the effects that we see there, to assume that they're going to work on the biology of aging to delay the predisposing biology of aging that creates a permissive physiology for manifestation of disease. So you talked about, kidney disease and heart disease. I'm not super familiar with this literature, so feel free to correct me if I'm wrong. I'm guessing, though, that those are in populations that had been diagnosed with at least a functional decline, if not with the disease itself, before they were given the medication, right? And so I think we just have to be a little bit careful not to assume that a drug that's going to work in a patient population where there's a pathology, an existing pathology, is necessarily going to work on the biology of aging to reduce the risk of developing that pathology as a function of age. It may, but it certainly doesn't have to. And so that's kind of where I would put canagliflozin at this point. I agree. It's absolutely exciting because we know, kind of like rapamycin, although in this case, I would guess the reputation isn't as much of a problem. We kind of know a lot already about its use in people and what the risks look like. We're starting to get a feel for what the rewards might look like. And so you can actually do a more informed risk-reward analysis when you've got that kind of information.
On the point of metformin, because I've been creating quite a bit of content, at least this year, around metformin, or about metformin, and about how, we've got fantastic research from the Diabetes Prevention Program where they gave metformin to non-diabetics, but they were high risk. So they had high, fasting blood sugar levels and they were overweight. And it was placebo-controlled, and we've got research now going, that data was followed up for about 15 years, and there was no health span or lifespan benefits seen. And in contrast, we've got research now showing that metformin seems to inhibit the positive effects of exercise, or at least halves the positive effects of exercise. So I've been mentioning to a lot of people who ask me either in the clinic or online whether they should be taking metformin if they're non-diabetic. And I say no, based on this, the risk-reward simply isn't there. Now, I prescribe metformin every day because I see type 2 diabetics every day. And for that population, absolutely, metformin is brilliant. But for non-diabetics, we certainly need to tread cautiously because there's a strong possibility that we could be causing more harm than good. And that's what I see online, where people that they try these new possible therapies or diet interventions that they think will actually improve their health. So what do you think? How can people, if they're trying to take health into their own hands, what's your opinion about how people can actually navigate this online world to make sure that they are doing good health decisions?
Yeah, super important question. Let me just make a comment about metformin, and then I'll answer the question that you just asked, because I think, again, metformin is really interesting, the way this has sort of evolved over the last 5 years or so, right? I think it was probably about 5 years ago when, it started to become popular in the biohacker community and a lot of people started taking it. And the perception then was that, well, metformin's great because it's got no risks. I mean, that was the perception. That's not the reality. The perception was it's got no risks and it might be helpful. And I think what we're learning is that the risk profile of metformin is actually potentially much higher than we first appreciated. The comments you made about impairing the benefits of exercise, again, I'm not completely convinced about that data, but absolutely there is evidence that, at least in some context, metformin can attenuate some of the beneficial metabolic responses to exercise. I think, again, exercise is kind of a catch-all term, so maybe it depends on the type of exercise, intensity of exercise, all of that. But yes, there is reason to believe, at least in some people, metformin may counteract the benefits of exercise. The thing I didn't even realize until relatively recently, there's a significant fraction of men when they take metformin where their testosterone drops quite substantially, right? That's a pretty big side effect that nobody ever talks about. They don't talk about it because I guess if you're diabetic, you're willing to accept that, to attenuate your diabetes. But in people who are not diabetic, I'd be pretty worried about that, especially if I was a man, right? So, and I am. So I think that, this idea that a drug is safe and there's no risk is that's almost, if not never, going to be true. And I think we're learning that the risks associated with metformin are actually much greater than was the perception a few years ago. So that's I also agree with you. Like I don't take metformin. I never have, and I wouldn't recommend it to people who don't have diabetes. Maybe if you're pre-diabetic, but I again, talk with your physician about that. Don't just go off and start taking metformin would be my advice. Okay, so I think your question was, how do people navigate the noise, basically? There's a lot of noise in the wellness world, a lot of noise in the longevity world. Everybody's out there trying to sell you something or make recommendations. It's really hard. So I think the one thing I would say is if somebody is telling you with confidence that anything other than a healthy diet, exercise, and sleep are going to benefit you for longevity, be skeptical. We don't have a ton of data to support any intervention for health span or lifespan in humans. There are things you can point to where there's some evidence, but I think that responsible people talk about the uncertainties associated with those interventions. So there are lots and lots of people saying, you should take this to reverse aging. I hate that term, by the way. Or, you should take this for longevity. It's mostly snake oil. Some of it, there's some evidence to support, but if they're telling you you should do that, they are not being honest with you about the uncertainties around whether or not that intervention works in people or is likely to work for you. So unfortunately, that's not what most people want to hear, because they want to be told what to do, right? But the reality is, this is a lot of educated guesswork. And the people who aren't being honest about the fact that it's guesswork are usually trying to sell you something and take your money, not help you. So that would be my advice for people.
To push back on that, creatine. So I think virtually everyone should be taking creatine outside of people who have got severe kidney disease. Because creatine seems to be fantastic for not only muscle recovery, but also muscle performance in combination with exercise. And when taken at reasonable dosages, so I'm talking about 2.5 to 5 grams a day, it seems to be extremely well tolerated, and we've got a truckload of human safety data now going out about 5 years, virtually dispelling all of the previous held notions around creatine, in particular, again, the kidney side effects. So, what's your opinion about creatine?
So I don't have a strong opinion about creatine, to be honest with you. So I'm certainly not going to disagree with your opinion, right? I mean, I think that's fine, and I think it's based on data, right? So again, I think what's important though is to be honest that we don't know with certainty what percentage of people would actually benefit from it, what lifestyle they need to be living to benefit from it. I'm very doubtful that somebody who is eating, a very high calorie diet, high in simple sugars, sedentary, is going to get any benefit from taking creatine. Maybe, but I'm very doubtful. So I would say, and you said this, right? You said in combination with exercise resistance training, right? So I think that's important. I think also, I think maybe the message needs to be, I don't think any of these supplements are going to move the needle at all if you don't have the foundation in order, right? If you don't have the healthy diet, or at least a reasonably healthy diet. I would say you don't have to go over the top and trying to be perfect. You shouldn't go over the top and trying to be perfect. But if you're not getting to a normal weight and you're not active, I don't think any supplement stack is going to help you substantially. It might, but I don't think there's any reason to think that it will. So get your foundation in order first, and then if you want to try to optimize on top of that, great. And again, then it comes a risk reward, right? So right, creatine, low risk, right? Potential reward in that population, great. If you want to recommend it for that population, great. But I think it's just important to be honest about what we really do know. And I mean, you do this, right? I think you should be commended for this. You're willing to publicly change your views if new data comes out. And I wish more people would do that. I think too many people get into their own biases, and data comes out that argues against those biases, and they just ignore it, right? Because it doesn't fit their worldview. So I think that's another important thing to help the general public navigate the noise is, if you're going to take advice from people, take advice from people who, number one, are honest that this is an educated guess right now and are open to changing their educated guesses when new data suggests that that's what they should do.
Yeah, I completely agree that it's the foundation that really matters. And again, that's one of the things I often talk about on the channel, that, diet, exercise, and sleep, they always come number one. And anything that we consider needs to be in addition to that, certainly not a replacement for that. So on that point, best exercises for longevity.
Anything is where I would start. So again, this gets back to the foundation, right? Doing anything active is better. Well, okay, almost anything active is better than doing nothing, right? So that would be the first comment. So my personal view. I mean, again, it depends on how far into the weeds you want to get. I think for most people, unless you're trying to really optimize your physical health through exercise, or you're a competitive athlete, for most people, you don't really have to think a lot about this. You want to do a mix of resistance training and cardiovascular exercise. And you want to do something that is sustainable for you, right? And so that means there has to be some degree of a fun factor, or at least it doesn't make you miserable, right? So my personal view is, I think this doesn't work for everybody, but I think everybody would benefit from just the basics on resistance training. Get some free weights, get in the gym and do the basic compound movements. Those work. I think that, again, not everybody does that. Some people are intimidated by that kind of thing. And my advice would be get over it and just do it. Because I think once you develop the habit, it sticks with you. But if you can't do that, use whatever you need to use, maybe bands, whatever. Do some resistance training to maintain and hopefully build muscle. Combine that with a diet that's going to allow you to build muscle. And then get out and do something for cardio. I don't think you really, again, I think for most people, you don't have to worry so much about, intensity of training and all of that stuff, at least at first. Get out and walk. If you like to hike. If you like to ride a bike, ride a bike. If you like to get on a treadmill and watch TV, great. Just do something. I think for a lot of people, these fitness trackers, if having a goal of 10,000 steps works for you, great, do it. Right. So again, my feeling is don't overthink it, at least at first. Once you get to a point where you feel like you have the foundations of exercise in place and you're doing it consistently and you feel like you can stick with it, then if you want to optimize on top of that, great, do it. But I think for 99% of people, you don't really need to worry about what zone you're training in at first, at least. So that would be my advice. The other thing I want to say, because you said diet, exercise, and sleep are really the big three. I would actually say there's a fourth one there, and it's happiness. I think we don't spend enough time thinking about what makes us happy, and if you're not happy, or if I'm not happy, I'm not worried about living a long time, right? If you're miserable, what's the point? So again, I don't have the answers, but I would say if you haven't thought about what makes you happy, think about it, because I think that's super important for optimizing your health span, certainly. I don't know how much effect it's going to have on lifespan. You can find evidence that, happiness, mindfulness, your perception of stress does have an effect on longevity, but absolutely it's going to affect your quality of life. So I would put that as equal to, if not more important than the other three. And obviously they're interconnected, right? If you're unhealthy, if you're sick, it's much harder to be happy. But I think I would really like to see more people in the wellness community think a little bit more about how to help people who are listening to them be happy rather than, the alternative.
Yeah, I completely agree with all of that, in particular with the exercise comments that you made. When I see patients in the clinic, I want to make sure that they are doing a mixture of both cardio and resistance, and in such a small window in the day that I'm seeing them, so we only have 15-minute appointments, we don't necessarily have time to go into, what zone training you're doing and what particular resistance exercise you're doing. I just want to know that you are doing some resistance exercise and some cardio exercise. So I completely agree with that. What do you think about people that say we can solve aging, that we can cure it and hopefully even one day reverse it? Do you think that that's a realistic possibility, or do you think that it's more about trying to find things that will increase health span? Essentially what I'm trying to get at is, will we ever, quote unquote, solve aging?
Well, I never say never, right? I think we have to say, it is. So there's nothing biological that I can think of that says it's impossible to halt or reverse the biological aging process. So theoretically, sure, it's possible. Are we anywhere close to doing that? I think is a very different question, and the answer is no. So again, I sort of alluded to this previously. In laboratory animals, the most effective intervention for increasing lifespan and health span in terms of magnitude of effect is caloric restriction. You get about a 50%, 60% at the top end. That's a big effect, but that's the biggest we've ever done, and that's been known about since 1935. So why haven't we done better than that in terms of magnitude of effect? The things that rapamycin is probably, at least for non-genetic interventions, let me be precise. You can do slightly better if you combine specific genetic interventions, but about the same as caloric restriction. For non-genetic interventions, rapamycin is second best, and then it drops off real quick. So we don't have anything in a mouse even that's close to immortality, right? We don't even double lifespan. So I think we're far away from having a sufficient understanding of the biology of aging to being able to, massively, the sort of extreme lifespan extension. Doesn't mean it couldn't happen. Doesn't mean there couldn't be a breakthrough next week that would be a game changer. Some people think that epigenetic reprogramming is going to be a game changer. It might, but so far there's no evidence to support that in terms of real data. So my view would be, show me first you can double the lifespan of a mouse, and then talk to me about reversing aging or immortality. But show me first you can do the little baby step, which we haven't been able to do in the last century. So I think that's important just for people to appreciate. So I think that, again, I feel like part of the reason why we haven't done better than rapamycin in terms of drugs is because nobody's really looking. Everybody is focusing on these sort of incremental interventions that have, 5%, 10% effects, instead of really trying to look at what we don't know and identify the interventions with big effect sizes. Again, the one exception right now might be epigenetic reprogramming, but so far nobody has even shown you can extend lifespan in a mouse by epigenetic reprogramming. So I'd like to see people looking a little bit more broadly outside of what we know to find interventions of large effect size. Reversing aging, I think, is also an important term to discuss. So I think it gets misused and misunderstood a lot. So it is absolutely the case that nobody has reversed aging in mice, certainly not in people. And I don't know of a case in any animal where anybody has reversed aging where what you mean by reversing aging is making an animal with an old physiology have a young physiology again. Nobody's done that. What has been done are select cases where in a tissue or an organ, some of the molecular changes that go along with aging have been reversed, and some of the functional changes that go along with aging have been reversed. This gets talked a lot about, again, in the context of epigenetic reprogramming. So we know that with age, there are changes to the epigenome, specifically in this case, methylation marks that change with age, and that epigenetic reprogramming can reverse those methylation marks. Is that reversing aging? Did it make an old mouse young again? No. It changed one phenotype of aging, or sometimes multiple phenotypes of aging. If you really want to talk about reversing aging, rapamycin is the best candidate we have out there. And as I mentioned earlier, functionally in brain, heart, immune system, oral cavity, and female ovary, there's evidence that rapamycin can actually restore function that has been lost in an old animal. Did it make that old mouse young again? No. Did it take it back functionally in some of these tissues to a more youthful functional state? Yeah. So I think you can say that we have rejuvenated certain aspects of function with age in a mouse, but nobody has taken an old mouse and made it young again. And that gets unfortunately misrepresented in the sort of popular sphere and the media a lot. There was a very, I think, misleading article in CNN a few months ago talking about epigenetic reprogramming, and they showed, an old mouse, and then right next to it in a different picture, a young looking mouse, and said, aging has been reversed. And that's just bullshit. It wasn't. But it was put out there. And I think if you read the article, you would get that. You would realize that aging hadn't been reversed. But it was put out there for people to look at that picture and immediately assume that scientists had taken an old mouse and made it young again. And it's just, it's unfortunate. It's misleading. I think it's damaging to the credibility of the field.
Because I've only been in this community for the past 3 years, I'd say. So, my background is always enjoying sport as a kid, trying to, do as much exercise as I possibly could, taking creatine to help with that extra little gain, going through medical school, and then, discovering that there are a couple of, wrinkles around my eyes. What is happening about this process of aging? And just in that short 3-year time frame, there's been different things that people have been excited about, whether it be resveratrol or NAD precursors, metformin, senolytics. Now we're on epigenetic reprogramming. So I wonder if you could just speak a bit more about the epigenetic reprogramming, because that seems to be the thing at the moment. But I've not really come across much convincing research, and I think you've alluded to this, around the potential reversal of aging or other markers of health. So realistically, what is the potential, do you think, of this therapy, and what might be achieved through this types of research, such as, say, from Altos Labs?
Sure. So again, first, I'll talk about what you said first. So you kind of went through a list there of things that were popular in the field for a while, and I don't want to imply or leave it out there as the implication that those things are all equal. So I think you said resveratrol, NAD precursors, senolytics, and epigenetic reprogramming. There's a lot of potential, I think, for senolytics as a clinical, both as a concept, clearing senescent cells, and as a clinical tool to have an impact on health span, potentially lifespan in people. So I wouldn't want to lump senolytics in with, certainly not with resveratrol. And NAD precursors, I think, still open question whether there's anything real there in terms of the biology of aging of significant magnitude. Okay, so epigenetic reprogramming is I'd say there's a lot of potential. It's just unfortunate that the hype has gotten way ahead of the data that we've got right now. So first, the concept of epigenetic reprogramming. So when we talk about epigenetic, what we mean are changes on top of the DNA, the genome, the genetic makeup of an organism that control gene expression. And those there are several types of epigenetic marks that are known to control which genes get turned on and off. The one that most people talk about when they're talking about epigenetic reprogramming is methylation, but there's acetylation and ubiquitination. Those also are other types of epigenetic marks. Okay, so there's a couple things I think that led to the excitement around epigenetic reprogramming as a longevity health span intervention. One was the development of these epigenetic clocks, and Steve Horvath and others are sort of famous for creating these tools that measure the changes in the epigenome that occur with age. And this happens, as far as we know, at least in every animal where this has been studied. There are predictable changes in the epigenetic marks that happen with chronological age. You can actually, with a fair degree of precision, determine the chronological age of a cell or an animal based on the changes in epigenetic marks. So that's these epigenetic clocks. And that led to a hypothesis that the changes in the epigenome are actually they're the upstream drivers of biological aging. So if you look around the hallmarks of aging, everybody's familiar with the hallmarks of aging. Epigenetic changes is one of the 9 hallmarks of aging. But there's this hypothesis that it is the primary hallmark of aging that some people have. I don't believe that, but some people have proposed that it's really the epigenetic changes that are causing all of the other biological changes that go along with aging. So if that were the case, if you could reverse those epigenetic changes, you could reverse or at least stop biological aging, because epigenetic changes are driving all the downstream effects. So that's the concept of why reversing the epigenetic changes with age might be beneficial, might reverse aging. And then there was a series of studies showing that you could actually reverse the epigenetic profile of cells using these transcription factor is called the Yamanaka factor. So this was actually a Nobel Prize winning research. This was done in cells. And so we know that if you have what's called a pluripotent cell, which can turn into all sorts of different types of cells, that differentiation process is driven largely by epigenetic changes. So it's the changes in the epigenome that determine which genes get expressed, and then the genes that get expressed determine the cell type that cell becomes. And what was discovered was you could use these Yamanaka factors to reverse that process. Take a fully differentiated cell, reverse it back to a pluripotent state, and hypothetically that cell has become young again. And there's evidence consistent with the idea that some of the damage that accumulates during the differentiation process can be reversed. So you have the concept that the epigenome is upstream of everything else in aging, and you've got a tool in these Yamanaka factors that in theory you could use in an animal to test that hypothesis. And so people have started to do that. So people have started to transiently express the Yamanaka factors, or different variants of the Yamanaka factors, to partially reprogram the epigenome in tissues in mice. And the results so far, have been encouraging. So people have shown that you can in fact change the epigenome of an old mouse back to something that looks more like a young epigenome, and that you can get some functional improvements associated with that in a couple of different tissue types. What nobody has really shown, though, is whether or not those long-term effects last. Can it be done at the whole animal level to truly reverse aging, broadly speaking, in many different tissues and organs at the same time? Can it be done to extend lifespan? And are there side effects associated with reprogramming that offset the potential benefits? Those are all open questions that we don't know the answer to, even in mice. So I think that's kind of where the field's at right now. It's still a hypothesis that it's these epigenetic changes that are upstream of all the rest of aging. Again, I don't think that's true. It might be upstream of many of the effects of aging, but we don't know yet. And nobody has shown really that you can have a robust effect in mice from this sort of a transient reprogramming strategy. And I'm not being critical of people for having not done that. It takes a while. This is not easy. It takes a while to do this. What I am critical of is the overinterpretation and the hype that some of the scientists in this area are using for personal gain and attention. That, I think, is irresponsible and damaging to the field. So I'm cautiously optimistic. I am also somewhat doubtful that you're going to be able to do better than caloric restriction in terms of magnitude of lifespan effect with reprogramming. I hope I'm wrong. I'd love to be proven wrong. But my guess is you can get about as good as rapamycin. That's probably where we're going to see this sort of approach level out. That's my guess. Like I said, I hope I'm wrong, but that's kind of my guess as to how this is going to play out.
So last time I spoke with you, I asked you about what supplements you are personally taking, and I from memory, I think it was rapamycin and vitamin D. Has that changed at all?
No, I mean, again, I don't take rapamycin continuously. I cycle it. In fact, it's been a while since I've taken rapamycin. I'll probably be starting again in the next couple of weeks. And then, I think a daily multivitamin makes sense for most people. I don't take one every day, but I do, when I remember, to be honest with you. And vitamin D is kind of the same, kind of put it in the same category. So again, my feeling is, my feeling is, the foundation is really what's important. And I think that there are supplements. Maybe I'll try creatine based on your recommendation, Brad. See if I notice any difference. But my feeling is the foundation is what's really important. And if you really put your effort into diet and exercise and sleep and happiness, which I think is important, then maybe some of these supplements can be beneficial on top of that. I'm pretty skeptical of the supplement industry as a whole. And I think this is a real challenge. We talked about how do people navigate this, not just the longevity field, but the supplement industry as a whole. How do people navigate this? Because there's so much stuff you could take, and you're constantly being told, take this, take that, take that. I am extremely skeptical of the entire industry. There's very little good research done to really show which supplements are beneficial, who are they beneficial for. I think the personalized aspect here is really important. And so my intuition is that 99.9% of the stuff that's out there is not going to be substantially beneficial, even for people who have got diet, exercise, sleep, happiness, right? The foundation pulled together. And so, and I don't know which 0.1% is going to be beneficial. So I'm kind of waiting, to feel confident about what else might make sense. But I also would say, I don't think, even in people who've got the foundation together, and again, if you have a difference of opinion, absolutely let me know. But I don't think there's anything that really moves the needle a lot. There might be some things that move the needle a little bit. I don't think there's really anything that moves the needle a lot for people who get, that foundation under control. I think for some people, rapamycin could move the needle a lot. I mean, I think I told you previously about my experience with adhesive capsulitis or frozen shoulder. That was a specific quality of life condition that rapamycin moved the needle a lot, right? So I think there are specific cases where sometimes supplements or rapamycin can do that. But I guess I'm just not convinced that there's a lot out there that's going to be likely to have big effects. The other thing I think that's important to appreciate too is, a lot of people take 7, 8, 9, 10 different supplements at the same time. And we have even if you're convinced that the data is solid for a particular supplement, we have very, very little data on these combinatorial interactions. We talked earlier about rapamycin plus acarbose for longevity. I mean, the number of longevity studies that have looked at more than one thing at the same time in terms of interventions, small molecules, tiny. So we have very little data on how combining different supplements will interact, if you're even getting bioavailability in the human body. And so my intuition is that more often than not, in fact, we want to do this experiment in laboratory animals and find out. But my intuition is more often than not, these things are going to cancel each other out or maybe even become detrimental in combination. And so again, I personally feel like I want to be pretty sure that whatever I'm doing is that the risk reward is favoring the reward before I start doing it. And so that's my personal philosophy on why I don't take much, at least compared to a lot of people out there.
Yeah, so I do agree with what you said about how we don't have a huge amount of research looking at particularly the combination therapies. And I can't actually think off the top of my head about any supplement that I take that the intervention testing program has actually shown to extend lifespan. So for example, even though I've designed a study looking at rapamycin, I personally don't take rapamycin because I'm waiting for that human research to come through. The supplements that I do take, and I've mentioned creatine before, it seems because we've got that research, when you combine it with exercise, that it gives further beneficial effects. The other supplements that I do take, it's not necessarily to target aging per se, but it's certain health aspects. So for example, I take collagen peptides, and that's because of the human research that we've got looking at skin health. So I don't take it to try and reverse aging. I take it to maintain skin health. And if it's helping my skin, is it helping my joints? I don't know. We don't have that research, but it does seem to improve skin health. And it's a similar story for why I take hyaluronic acid. There's no research to suggest that it would improve my lifespan, but if it's going to improve my skin health, that's a supplement that's worth taking.
Yeah, I think those are interesting examples, though. And again, maybe you're more familiar with this research than I am, but like collagen peptides, if you think about the biological, so I'm not arguing that there's some evidence that it can improve skin health, right? I think that there is some evidence for that. But if you think about the biological mechanism, it doesn't make a lot of sense to me why collagen peptides would have that effect, right? So the way that these things are going to get into your gut, be acted on by the microbiome, potentially be taken up as, broken down peptides, it doesn't make a lot of sense that they're then going to be, resynthesized as new collagen, right? So what makes them special? There may be data on that I'm not aware of, but I tend to get very skeptical when the biological mechanism for how these things might be working doesn't make a ton of sense. Not impossible, but it doesn't really all hold together. I think the NAD precursors are another interesting example where, it kind of made sense. These things are precursors of NAD that you could take them, they would get taken up and you'd get more NAD. Now we're learning that they probably get broken down by the microbiome immediately, right? And then maybe it gets taken up as an earlier precursor and eventually gets brought back to NAD. But I would like to see, and this is part of the challenge with the supplements, because nobody is required to actually do the research and show how these things are working, if they're working from a biochemical perspective before they start selling them. I would really like to see more responsible research actually showing a plausible biological mechanism, bioavailability for the outcome that is claimed, right? And that just seems to be missing. And again, like I said, I have trust issues with the supplement industry. On the other hand, they're doing what they should do as companies whose goal it is to make money. They're not required to show that these things work. They're not required to show how they work. That's a problem with the system and the way that we regulate supplements. And I think the other thing I guess is also worth saying is, some people get a pretty profound placebo effect from supplements, and that's okay, right? Placebo effect is real. And if you're getting a perceived benefit, great. I do wonder though, how many people and what the economic cost is sometimes. And, could you be spending that money on stuff that might be better for your health? I don't know. Like I said, I have a hard time with the supplement industry as a whole. And I think, thinking about some of these specific supplements and what that mechanism of action could be is part of the reason why I'm skeptical, I guess.
Yeah, I think let's focus in on collagen peptides just in the couple of minutes that we've got left, because I think this is a really interesting case. So the reason why I take it is primarily because of a 2020 meta-analysis that included 10 separate human clinical studies that were all placebo-controlled, and they were double-blind. And these studies were specifically looking at outcomes of skin health, so to do with, wrinkles, skin elasticity, hydration, and all 10 of those studies, again, that were placebo-controlled, showed improvements in skin health. And obviously when combined in that meta-analysis, you can see that effect. So that's the reason why I take it. Now, that paper speculated about 3 possible mechanisms as to how these peptides may improve aspects of skin health. And one of the mechanisms was to do with this idea that maybe the immune system is sampling the contents in the gut. So if it can detect these collagen peptides, maybe there's less breakdown of collagen in your skin. But the overall conclusion was that we're not sure, but we do see an effect. Hence why I take it. So again, I think it's reasonable to take the collagen peptides because of that study, making sure again that none of this replaces diet, exercise, sleep, and as you've mentioned, happiness.
Yeah, and I think that's a reasonable sort of risk-reward analysis, right? Or thought process for why you take it. Again, I think the one thing I would say about those sorts of epidemiological studies is first of all, 10 years from now, we might get the opposite result. But that's not a reason not to believe them. I think you just have to be aware that oftentimes these things are problematic, even when it's a meta-analysis and you find out later that for whatever reason it wasn't real. I think the more likely thing, though, and this is where, having a better mechanistic understanding could be important, is even in these epidemiological studies, you usually get small effects, and what never gets talked about are responders and non-responders. And so for a lot of these things, we don't know who actually were the people who got the benefit in that study. Maybe everyone did, or maybe it was a small percentage of people who maybe were eating a low protein diet and taking these peptides. I'm totally speculating. I have no evidence for that, right? But I think these are all part of the reason why it's so hard to have a lot of confidence in a lot of these studies, even when there is some epidemiology to support it. Having said that, like I said, I think your thought process is perfectly reasonable. And again, what's the downside? There probably isn't any significant downside, at least as far as we know, other than, the cost of the supplement. And I actually don't know how much collagen supplements cost. So I don't know if that's significant or not. For some people, it'd probably be significant. But those are the kinds of things that trying to do that, and you have the ability to actually look at the data and do that sort of, thoughtful analysis. I think most people don't. And this gets back to that noise issue of who do you believe? There's a lot of people out there telling you what to take. It's really hard to know who to believe.
Matt, if people want to know more about you, where should they look?
Yeah, so Dog Aging Project is dogagingproject.org. We are absolutely still recruiting dogs, all sizes, shapes, weights, ages. So if you have a dog and you're interested, please go to the website, nominate your dog. I'm on Twitter at @mkaeberlein. My lab website is kaeberleinlab.org.
It's been an absolute pleasure. Thank you very much.
Thanks, Brad. It's been fun.